Okay, this video is on microbial genetics, and this lecture is going to be a little bit different from the other ones. This is from a different textbook, and I think that it's got a lot more information that I want to really use here. So this is probably one of the most important chapters we'll have in this class. Um, and so we really want to dive into this. So you guys have learned about genetics. We're going to bring that in, but we're really going to focus on bacterial genetics, and especially these three different types of horizontal transfer. So we have transformation, transduction, and conjugation. But before we get too crazy, let's talk about what genetics is. So genetics is the study of inheritance or heredity, um, that explores the transmission of traits from parent to offspring, how those traits are expressed, the structure and function of genetic material, and how this material changes. So at the organismal level, you see an actual physical characteristic. At the cell level, you see the chromosomes, or the cell. And then chromosome level, you've got eukaryotes and prokaryotes. And then at the molecular level. So we're going to dive all the way in. The genome is the sum total of all the genetic material of an organism. Most exist in the form of chromosomes. Some may appear in non-chromosomal forms, like a plasmid is a tiny extra piece of DNA. And organelles, actually, there's two that have their own DNA due to that endosymbiotic theory, where they believe that the mitochondria and the chloroplast may have been a bacteria at one time. There's also genomics, which is the study of an organism's entire genome. So this just looks at the general location. So with a eukaryote, the chromosomes or the DNA stay inside of the nucleus. They never leave. RNA can leave the nucleus. DNA can be in chloroplast and mitochondrion. And some fungi and protozoa have plasmids, whereas bacteria do not have a nucleus. They have a nucleoid, and they have a circular chromosome, and then they might have plasmids. Viruses, we talked about this in the virus chapter, where we've got DNA and RNA viruses, and there's lots of different types there. So at the level of structure and function of the genome, so a chromosome is a discrete cellular structure composed of a neatly packaged DNA molecule. Eukaryotic chromosomes are located in the nucleus. They vary in number from a few to hundreds. If they occur in pairs, they're called diploid. If they occur in singles, they're called haploid, and they have a linear appearance. Whereas the bacterial chromosome is usually singular, circular, double-stranded chromosome, although many bacteria have multiple singular chromosomes, can have some multiple, or they can have some linear. Genes are the basic informational packets. Classical genetics is there's a functional unit of heredity. Molecular and biochemical genetics, the site on the chromosome that provides information for a certain cell function. A preferred definition is a segment of DNA that contains the necessary code to make a protein or an RNA. The genotype is the sum of all types of genes constituting an organism's distinctive genetic makeup. So that's the genes. And then the phenotype is the physical characteristic of that gene. So the expression of that genotype will make certain structures or functions. Size and packaging of chromosomes, so variation in genome size. So E. coli has a single chromosome with about 4,000 to 5,000 genes. It's about 1 millimeter long if unwound and stretched linearly, and it's 1,000 times longer than the cell, whereas a human cell has 23,000 genes on 46 chromosomes. So that's why we're more complicated. This is just showing an electron micrograph of DNA. Structure, we've talked about this before, but we'll just quickly go through it again. Basic unit of DNA is the nucleotide. We have a phosphate, deoxyribose sugar, a nitrogenous base. You have purines and pyrimidines, so A always pairs with T, and G always pairs with C. It looks like this. You've got covalent bonds along the outside with those phosphodiester bonds, and then in the middle you have hydrogen bonding. And the hydrogen bonding is very important because it will unzip, and that's how we can replicate. Another important characteristic of DNA is its anti-parallel arrangement. So on one side of the helix runs in the opposite direction of the other, so 5' to 3' in one direction and 3' to 5' in the other. This is a significant factor in DNA synthesis and protein production. So you're going to hear me say 5' to 3' and 3' to 5' a lot today. So these are the simplified steps of semi-conservative replication. And so you've got the origin. And so it is going to go in two directions. It's bidirectional. So you've got the leading strand, which is going to be the proper way that it's going to function. And then you've got the lagging strand, which is going to be little pieces. And those are called Okazaki fragments. And the DNA polymerase is going to go in those different directions. So some enzymes you should know. A helicase is going to unzip the helix. Primase is going to synthesize the RNA primer. DNA polymerase 3 is going to add bases to the new DNA chain, proofreading the chain for mistakes. DNA polymerase 1 is going to remove the primer, close the gaps, and repair mistakes. Ligase is going to will bind the nicks in the DNA during synthesis and repair. Topoisomerase 1 is making single-stranded DNA breaks to relieve the supercoiling. And then topoisomerase 2, or also called DNA gyrase, and topoisomerase 4 are going to make the double-stranded DNA breaks to remove the supercoiling ahead of the origin and separate the replicated daughter DNA molecules. Overall replication process is semi-conservative. That means each daughter strand is identical to the parent in composition. Neither is completely new. The template strand is the original parental DNA strand. DNA polymerase 3 is going to synthesize a new daughter strand of DNA using the parental strand as a template. The DNA molecule must be unwound and separated before the DNA polymerase 3 can function. It can only add to an existing chain. It cannot begin synthesizing without the nucleotides. Can only add to the 5' to 3' direction. The replication fork is the place in the helix where the strands are unwound and replication is taking place. Each circular DNA molecule will have two identical replication forks. The primer is a length of RNA that is inserted initially during replication before being replaced by the DNA. Some other terms, you've got the leading strand, which is the strand of new DNA that is synthesized continually in the 5' to 3'. The lagging strand is a strand of DNA that must be synthesized in short segments because it's going the opposite direction. And that is, it's going opposite of the 5' to 3', but the polymerase only goes to that. So it's going to hop off and go back and forth. Later, it's sealed together to form the 3' to 5' strand. The Okazaki fragments are what those are called. Those little short segments of DNA are called Okazaki fragments. So this is showing the DNA replication. So you've got the topoisomerases are going to unwind. Helicase is going to break the hydrogen bonds. The single-stranded binding proteins are going to keep them apart. And then the DNA polymerase 3 is going to hop on and make this complementary strand. There are several videos that are in the announcements. So just so you know that that's coming up. So this is showing elongation and termination of the daughter strand. So this is showing the replication fork of a bacterial chromosome. So it's going to fold over like this, and then it's going to create a nick to separate the two circular strands. Replication of the linear DNA is similar to the bacterial and archaeal. It uses a variety of DNA polymerases. Replication proceeds in both directions, but from multiple origins. Topoisomerases are used to relieve tension in the strand and to recompact the molecule when it's fully replicated. So these are the videos that I was talking about. I am going to let them play. The The sound I don't think will come through, so that's why I put them there. But I want to kind of walk you through them, and I can talk you through them. So this is showing the parental strand.
In order to pass genetic information on to its offspring, an organism must make a copy of its DNA.
So it's
The
making a
process
copy
of
of the
copying
DNA,
DNA is called DNA
and that is
replication.
called
During
replication.
replication, each strand of the parental DNA serves as a template in the creation of new DNA. Because each newly synthesized DNA molecule is made up of one parental strand and one new strand, DNA replication is described as semi-conservative. One strand in each molecule is conserved, while the other strand is newly replicated.
Okay, so it was talking about that semi-conservative replication. So then this is going to proceed talk about the replication fork, so you can kind of watch and read, and then also watch these in the some more.
Enzymes called DNA helicases
So this is
use
showing the
energy from
stability
ATP to
proteins
break the
and the
hydrogen
DNA
bonds that hold
helicase.
the two strands of the DNA molecule together, causing the DNA to unzip. The site where DNA helicase unzips the DNA is called the replication fork. As replication continues, these enzymes continue to break hydrogen bonds, and the replication fork moves ahead of the newly synthesized DNA. Stabilizing proteins bind to the single-stranded DNA, holding the strands apart and protecting them from degradation.
Okay, and then the next video is about the proteins involved.
DNA polymerase can only add bases to the 3' end of the nucleic acid, which means DNA polymerase can only add bases to a nucleic acid. It cannot start replication from scratch. Cells use an enzyme called primase to synthesize a short piece of RNA called an RNA primer. This primer provides a 3' end on which DNA polymerase can act. An enzyme called DNA polymerase catalyzes the addition of bases to the 3' end of the growing DNA molecule. This reaction is powered by the energy in the high-energy bonds of the nucleotide triphosphates. Hydrogen bonding between the parental strand and the nucleotide triphosphates dictates which base is added. Only thymine fits correctly across from adenine, and only cytosine fits correctly across from guanine. An enzyme called RNase removes the RNA primers, leaving a gap in the DNA which can be filled by DNA polymerase. DNA ligase seals the maps between the DNA fragments created during synthesis of the lagging strand.
Okay, so that's showing the leading and lagging strand. So this idea of the central dogma. So the central dogma begins at transcription and includes both transcription and translation. So transcription is when DNA is synthesized into RNA or the transcript. And then translation is we take RNA and make it into protein. So the central dogma is actually incomplete because there's a wide variety of RNAs that are used to regulate gene function, and many genetic malfunctions that cause human diseases are found in the regulatory RNA, not in the genes for proteins. So the DNA that codes for these crucial RNA molecules was once called junk DNA, but it's probably not anymore. And so they're still investigating that. So DNA, the transcript can make tRNA, mRNA, rRNA, and then those can also make microRNAs, interfering RNAs, antisense RNAs, riboswitches, and regulate transcription and translation. So this is going to then lead to translation, where the ribosome hops on that rRNA, and protein will translate the mRNA using tRNA, and then you get a new protein. Oops. The gene protein connection. So there's a connection between DNA and an organism's traits. So a protein is the primary structure that determines its characteristics, shapes, and functions. Proteins ultimately determine the phenotype, and we can study something called proteomics. This is the study of an organism's complete set of expressed proteins. DNA is mainly a blueprint that tells the cell which kinds of proteins to make and how to make them. So they come in these triplets or these codons. So each triplet is part of a protein that codes for an amino acid. So you have the DNA that's a triplet, the codon is the mRNA, and then the amino acid comes from that. The major participants in transcription and translation, you've got messenger RNA, transfer RNA or tRNA, ribosomal RNA, microRNAs, ribosomes, several types of enzymes, and many raw materials. So this is showing the tRNA and the codon here. So I really like this figure. I would definitely screenshot this, and this kind of goes through all the things, all the different RNA types, all the descriptions, all the functions, and whether it's translated or not. So the differences in RNA structure. So it's single-stranded and it exists in a helical form, can assume a secondary and tertiary levels of complexity. It contains uracil instead of thymine as the complementary base pair to adenine, and the sugar is ribose instead of deoxyribose. mRNA is a transcript of structural genes or genes in the DNA, synthesized in a process similar to the synthesis of the leading strand during DNA replication. Codons are a triplet, and it will hold a message for the transcribed mRNA. The transfer RNA or tRNA contains the sequences of bases that form hydrogen bonds with complementary sections of the same tRNA strand. The anticodon is found at the bottom of the loop of the clover leaf, and it designates the specificity of the tRNA and complements the mRNA codon. The ribosome or rRNA is a large polynucleotide molecule, forms the complex three-dimensional shapes that contribute to the structure and function of ribosomes. So there are, depending on if you have a prokaryote or a eukaryote, they are the same, they look the same, just the prokaryotic ribosome is a 70S, whereas a eukaryotic is 80S. They interact with the rRNA and protein to create two subunits of the ribosome that engage in the final translation of the genetic code. So you have your amino acyl side, your peptidyl site, and then your exit. So it goes in. This is where your tRNA comes in. It makes the peptide bonds in the P site, and then it leaves the E site. Transcription starts with initiation. So I know we're kind of backing up a little bit because we've got to get the transcript first. Starts with initiation. And there's going to be some videos here. And then it goes to elongation and then termination. So here's one of the videos. And then we'll go through that.
Cells use DNA to store information, while proteins are used to perform the activities necessary to keep the cell alive. In a process called transcription, the information stored in a DNA molecule is copied into RNA molecules, which can be used to synthesize specific proteins. Cells use three types of RNA: ribosomal RNA molecules, which are used to form ribosomes; transfer RNA molecules, which deliver amino acids to ribosomes; and messenger RNA molecules, which carry the information for making specific proteins from DNA to ribosomes.
Okay, so that's just an overview. And then this one gets a little bit more into the actual process. And again, these videos, so you can listen to them, are on the SMORE. Most of them are, not all of them, but the important ones are.
There are three stages in the process of transcription: initiation, elongation, and termination. Let's see how each stage works.
So I'm going to skip through this because you can watch this. Just know that you need to watch this. This one's kind of long. So the master genetic code, or the message in the messenger RNA, is the central principle of translation. So you get the transcript and then you get the then you make the protein. So the mRNA nucleotides are written codons or groups of three. The codon dictates which amino acid is added to the growing chain. Except for a very few cases, this code is universal for archaea, bacteria, eukaryotes, and viruses. So there's a redundancy of the code, which means that certain amino acids are represented by multiple codons and allows for the insertion of the correct amino acid even when mistakes occur in the DNA sequence. There's a wobble. So the first two are required to encode the correct amino acid. The third does not change its sense. So it will make multiple amino acids on that third base. Permits some variation or mutation without altering the message. So you have the triplets, the mRNA codon, the tRNA, and then the protein. They all start with the codon AUG, which is the tRNA for methionine, and for DNA, it's formal methionine. But they all start with methionine. So translation is the second stage. It has the elements that are needed are the mRNA, the tRNA, the amino acids, and the ribosomes. It has 3 stages, just like transcription: initiation, elongation, and termination. The start codon is the first 3 RNA nucleotides that signal the beginning, which is always AUG. There's also a stop codon, which is nonsense codons. There's UAA, UAG, and UGA, and that terminates or stops translation. Translocation is the process of shifting the ribosome down the mRNA strand to read the new codons. So post-translational modifications, so this is after the protein is made. So they start to fold upon themselves to achieve their tertiary conformation, even before the peptide chain is released. Formal methionine may be clipped off, cofactors may be added, and some join with other proteins to form a quaternary structure. So the thing that's really interesting about bacteria is that they kind of form an assembly line. And so this shows that you can have transcription and translation occurring at the same time. So multiple ribosomes are going to hop on there and start making those proteins as soon as that transcript is made. So you can see it's a polyribosomal assembly line of proteins. So the differences between a prokaryotic and a eukaryotic transcription and translation is that co-translation is only in bacteria and archaea. AUG codes for a different form of methionine in eukaryotes, and eukaryotic mRNAs only code for one protein, whereas bacterial mRNAs may contain information for several genes in a series. Another thing is that eukaryotes have to go through a post-transcriptional modification before they can be translated. So what happens here is most eukaryotes don't exist in an uninterrupted series of codons. So what has to happen is the introns don't code for protein. That doesn't mean they're not important, but they just don't code for the protein that's needed. So those are excised or taken out before they can be made into that transcript that can be translated. So the mRNA is just a series of exons, which are the coding regions for the proteins. And this is showing the spliceosomes that actually take those little pieces out. The spliceosome will recognize the exon and introns and will use enzymes to cut through them. Loops in the introns they loop them into lariat pieces and excise them, and then the complete mRNA strand is then ready to be translated. Genetics and animal viruses. So viruses consist of one or more pieces of DNA or RNA enclosed in a protective coating. So they are genetic parasites that require access to the host's genetic and metabolic machinery to be replicated, transcribed, and translated. Also have the potential for genetically changing the cells they infect. They tend to be more compact and economical. The diversity of viral genetics, so some are linear, some are circular. And remember back in the genetics, the viral lecture, we talked about double-stranded DNA, single-stranded RNA, single-stranded DNA, and double-stranded RNA, and also the retroviruses. Okay, so this part is super duper duper important. So operons. I'm going to stop and create a new video because I want to make sure that this doesn't get lost in the video.